class, let's continue to study the eukaryotic cells and microorganisms. Last time we have learned about the characteristics, shape and size, cell organization of eukaryotic cells. In this lecture, we are going to learn about the structure of eukaryotic cells in detail. The figure shown here maps the structural organization of eukaryotic cell. In general, eukaryotic microbe cells have a cytoplasmic membrane, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and glycocalyx. A cell wall, locomotor appendages, and chloroplast are found only in some groups. Most eukaryotic cells have cell walls, although they are generally much simpler than those of prokaryotic cells. The cell walls of the fungi and algae are rigid and provide structural support and shape, but they are different in chemical coloration from prokaryotic cell walls. First, many algae have cell walls consisting of the polysaccharide cellulose. Other chemicals may be present as well. Second, cell walls of some fungi also contain cellulose, but in most fungi, the principal structural component of cell wall is the polysaccharide chitin. Third, the cell walls of yeast containing the polysaccharides glucan and mannan. Fourth, in eukaryotes that lack a cell wall, the plasma membrane may be the outer covering. However, cells that have direct contact with the environment may have a coating outside the plasma membrane. Fifth, protozoa don't have a typical cell wall. Instead, they have a flexible outer protein covering called a pellicle. As shown in figure, many eukaryotic microbes, as well as cells of multicellular eukaryotes, have integral membrane proteins and lipids decorated with carbohydrates. This often forms a carbohydrate-rich layer on the surface of the cell called glycocalyx. Most eukaryotic microbes have a glycocalyx, which comes into direct contact with the environment. Some of these carbohydrates are covalently bonded to proteins and lipids in the plasma membrane, forming glycoproteins and glycolipids. Glycocalyx promotes adherence to environmental surfaces and the development of biofilms and mats. It also serves important receptor and communication functions and offers some protection against environmental changes. The cytoplasmic membrane of eukaryotes is a lipid bilayer composed of a high proportion of sphingolipids and sterols, in addition to the phospholipids observed in bacterial membranes. As shown in figure, A is the molecular structure of phosphatidylcholine, which is a kind of phospholipid. B is the molecular structure of sphingomyelin, which is a kind of sphingolipid. C is the molecular structure of cholesterol, which is a kind of sterol. Large amounts of sphingolipids and sterols contribute to the strength of the plasma membrane. They are able to pack together very closely, more so than phospholipids can. This explains in part why so many eukaryotic microbes are able to resist osmotic pressure and other mechanical stresses even though they lack cell walls. As with the plasma membrane of bacteria and archaea cells, eukaryotic cells must be able to move materials across their membranes. Substances can cross eukaryotic and prokaryotic plasma membrane by simple diffusion, facilitated diffusion, osmosis, or active transport. Group translocation does not occur in eukaryotic cells. However, eukaryotic cells can use a mechanism called endocytosis. Cilia and flagella are the most prominent external structures observed on eukaryotic cells. They are associated with motility. Motility allows a microorganism to locate life-sustaining nutrients and to migrate toward positive stimuli. It also permits a void of harmful substances and stimuli. Locomotion by means of flagella is common in protozoa, algae, and a few fungi and animal cells. Cilia are found only in protozoa and animal cells. As shown in figure, A is a micrograph of Euglena with its flagella. B is a micrograph of tetrahymener with cilia. Although both are whip-like and built to move the microorganisms along, they differ from one another in two ways. First, cilia are typically only 5 to 20 microns in length, whereas flagella are 100 to 200 microns long. Second, their patterns of movement are usually distinctive. As shown in figure, flagella move in an undulating fashion and generate planar or helical waves, originating at either the base or the tip. If the wave moves from base to tip, the cell is pushed along. A bit traveling from the tip toward the base pulls the cell through the water. Cilia, on the other hand, normally have a bit with two distinctive faces. In the effective stroke, the cilium moves through the surrounding fluid like an oar, thereby propelling the organism. The cilium next bends along its length while it is pulled forward during the recovery stroke in preparation for another effective stroke. Despite their differences, cilia and flagella are very similar in outer structure. They are membrane-bound cylinders about 0.2 microns in diameter. As shown in figure, located in the matrix of the organelle is the axonomy, which usually consists of 9 microtubule doublets arranged in a circle around 2 central microtubules. This is called the 9+2 pattern of microtubules. A barrel body lies in cytoplasm at the base of each cilium or flagellum. It is a short cylinder with 9 microtubule triplets around its periphery and is separated from the rest of the organelle by a basal plate. As shown in diagram, the axonomy consists of 9 microtubule doublets arranged in a circle around 2 central microtubules. Each doublet has pairs of arms projecting from subtubule A toward a neighboring doublet. A radial spoke extends from subtubule A toward the internal pair of microtubules with their central scenes. These microtubules are similar to those found in cytoplasm. Cilia and flagella bend because adjacent microtubule doublets slide along one another while mating their individual lengths. The doublet arms, about 15 nanometer long, are made of the protein dynein. Dynein arms interact with the B subtubules of adjacent doublets to cause sliding. The radial spokes also participate in this sliding motion. ATP powers the movement of cilia and flagella. Well, let's stop here for this lecture.